Hi, I'm Peter Balk. I'm the coordinator of the AppleGenie project. This project is funded by the European Communion. Within this project we are studying the possibility to use gene activity measurements to assist thinning practice and to prevent bitter pit disorder for the major apple varieties Golden Delicious, Red Delicious and Gala.
Among the techniques that can be used in an apple orchard by growers, thinning is one of the most efficient. Successful thinning makes apple growing profitable not only the year it is carried out, but also in the following years by minimizing biannual bearing. This operation has several effects which influence both quantitative and qualitative components. The decrease in crop load leads to an increase in size and an improvement in quality and color. Even if the technique is effective in reducing load, it appears difficult to manage. Chemical thinning is not an exact science. Several factors interact and require some adjustment for each orchard. Currently, there is no reliable model for predicting thinning response, and there is no decision-making tool to help growers to build their thinning program. The goal of the AppleGenie project is to read the status of the tree directly by molecular analysis. This method will avoid having to consider numerous factors involved in making an actual decision. The status of the tree will be assessed once before the first spray to define tree abilities to feed fruits. A second assessment will be done 3 days after the spray to predict thinning response and decide if an extra spray has to be done.
My name is Joan Bonani and I'm director of Virta Mas Badia. Today is to talk about bitter pit. Bitter pit is a physiological disorder that is affecting apple fruit. We know that it's a calcium-related disorder that has other type of physiological disorder. The most cultivated apple varieties like Golden and Red Delicious are sensitive to this disorder, and we know that it's also causing economic losses, important economic losses to many fruit growers around the world. The way that the grower is coping for this disorder now is by intensive applications of calcium products that go from right after fruit bloom until harvest. Another way to cope with the problem is trying to have tools that predict the incidence of bitter pit in a certain orchard. So then, if we could know it before harvest or right after harvest, that specific lot of fruit could be managed accordingly. Until now, the methods that we have had have not proven to be very reliable in predicting the final bitter pit incidence. That's why we got interested in other methods like the genomic tools, and these genomic tools are those that we are testing and we are trying to develop under the AppleGenie project.
Hi, my name is Peter and I work for the New Zealand Institute for Plant and Food Research. Our role in the AppleGenie project involved the analysis of gene expression to identify markers for thinning efficacy and bitter pit incidence in apple. We processed samples from Spain, Italy, France and New Zealand to get information from multiple locations and measured global changes in gene expression using a sequencing-based approach. We then looked for genes that showed changes in gene expression that were linked with thinning or bitter pit. Using this approach, we have successfully identified potential markers that could be used in a diagnostic tool and are currently testing how reliable these are. A second component of this project involved streamlining methodologies to analyze gene expression in the field. We are currently exploring new ways to sample, process and analyze tissue to facilitate a wider usage for this diagnostic tool.
Select 15 apples from one batch and cut a thin slice from every fruit. Take a small part of each slice, including skin. Put all 15 pieces in a single compartment of the bag. Add the liquid provided in the kit. Mash the apple pieces manually. For 1 minute, proceed immediately to step 6. Collect some juice using the pipette from the other compartment of the bag. Apply 2 drops of juice inside the circle of the sampling card. Let the card dry completely for 1 to 2 hours. Insert dried card into the little bag with drying agent. Now the card is ready to be transported to a lab for analysis of the RNA molecules from the selected indicator.
Hello, I'm Tania Marques and I'm representing Open Nature Company. I'm here on behalf of the companies that participate in the AppleGenie project. Our role in this project is to bridge the gap between science and end users. Practical value is assured by the companies. Cooperation between all the partners is perfect, and although the project is still ongoing, there are already promising results. This project is developing diagnostic tests to handle ineffective chemical thinning and the storage disorder bitter pit and overcome these problems in apple production. Project results provide valuable tools for customers, helping in decision-making to apply technology and achieving optimum productions. For further information, don't hesitate to contact us.
It's important for us to keep in touch with the end users to ensure that the tools used are relevant, science and practice. Perfect cooperation between partners. Contact us for any further information about this evolving project. We would be glad to explain more about the AppleGenie project. www.applegenie.eu, twitter.com/AppleGenieEU.